We're getting everybody joining online. Fantastic. Welcome, guys. Good morning. Good morning, everyone. Thanks for joining us. We have our Sea Grant Marine Mammal webinar. Just giving everybody a few minutes to log on. I think the numbers are still going up. We are so glad to have you this morning and have so many of you joining us. This is great. We are also streaming live on Facebook. So if you have any questions, please feel free to put them in the chat, the question and answers, or on Facebook. It looks like our numbers are pretty steady, so we can get started, I believe. I'll give it another 30 seconds. There's so many people joining. This is fantastic. It's so great to see y'all this morning. Okay, perfect. So I am going to introduce myself. I'm Dani Bailey. I work for Texas Sea Grant, and I am part of the Oil Spill Science and Outreach Program, which I'll get to in a little bit. Today, we're going to be talking about marine mammals. If you have any questions while we're going through the program, we're going to answer all of them at the end of this talk. So we're going to have 3 speakers and then a panel of 5 to answer all of your questions. So as we're going through, feel free to type your questions into the Q&A box. If you have any issues, please type everything or anything into the chat. And we will go ahead and get started. It looks like we're just about ready. So I'm going to share my screen. There we go. Oh, move it over here. Perfect. All right. Welcome everyone to our Marine Mammals Reflections on New Research After the Deepwater Horizon Oil Spill. So to start out with, I want to tell you a little bit about Sea Grant itself. So Sea Grant is 34 university-based programs. So anywhere there's a large body of water, Great Lakes, an ocean, we're there to help work with it. We are non-advocacy and science-based for our information. So we like to try and do as much research, education, and extension as possible throughout the communities that we work. And then we have 50+ years of serving the American coastline. So within that research, education, and extension that we do, we focus on healthy coastal ecosystems, sustainable fisheries and aquaculture, resilient communities and economies, and environmental literacy and workforce development. So those are some of our core areas within our Sea Grant programs. So if you ever need anything from Sea Grant, there's a Sea Grant near you, please reach out. I am part of the Gulf Sea Grant team. So as I said, I am Dani Bailey, and I work with all of these wonderful team members to make research or make outreach and information get spread throughout the Gulf. So we have our supervisor, Steve Sempier, who is in Mississippi, Alabama. We have Tara Skelton, who is also in Mississippi, Alabama. She's our wonderful communicator and having all the tinkering in the background behind the scenes. We have Emily Mong Douglas, who is in Louisiana, Monica Wilson, who's in Florida, and Missy Partika, who is in Mississippi, Alabama. So this is our team, and we work throughout the Gulf, coordinating outreach and information and webinars and sessions like this. So how we got started. 2010, there was the Deepwater Horizon oil spill. So BP gave $500 million to non-penalty funds. And from there, it was the Gulf of Mexico Research Initiative was formed and managed by GOMA. So it's a completely independent research board from any of BP management. It funds science and outreach. We try to keep that all science-based and not biased in any way. And then we have theme areas within this to study everything we could about the oil spill research. So we have fate and transport of the oil, chemistry of how it's all working together, environment and wildlife, technology, and all the aspects that go along with human health and our people that are using the Gulf as a healthy resource. So with that, a little bit about how we do our work is we're a neutral broker of information. And so we try to take government reports, peer-reviewed publications, and we'll turn those and translate those into outreach products, seminars, publications that can be used by anyone. So anyone who might be interested in some of these topics but doesn't have that science background, we try to be able to translate all of that information and the data and the findings that the scientists have found to everyone in the Gulf who might be interested, or outside of the Gulf. We've really worked with sort of an international team here. So with that, I'm going to talk about today's speakers. So we have Sarah Piwetz, Research Director at Texas Marine Mammal Stranding Network. We have Ryan Takeshita, Deputy Director of Conservation Medicine and a research scientist at the National Marine Mammal Foundation. And we have Sylvain De Guise, Professor, Department of Pathobiology and Veterinary Science, University of Connecticut. So with these three wonderful speakers, we are going to talk all about some of the research that has been discovered on marine mammals throughout these past 11 years and how that's really going to help us affect in the future. So I will stop sharing my screen and give a short introduction, if my computer screen will work. There we go. On Sarah. So Sarah is the research director at Texas Marine Mammal Stranding Network. She has nearly 20 years of experience studying dolphins, whales, including 6 species across 7 countries. The focus of much of her work is on the effects of human activity of coastal dolphin behavior and movement patterns, and she is broadly interested in behavioral ecology, stranding trends, and how human activity affects marine mammals. So with that, Sarah, if you could join us and share your screen.
Thank you, Dani. Let me get my screen shared up.
Absolutely.
Thank you for that nice introduction. And I will get right to it. Yeah, thank you for the introduction and for hosting this webinar overall. I think it's a great way to get the latest information. I've been asked today to talk about the Texas Marine Mammal Stranding Network and the role that we fulfill during oil spill response, the importance of stranding programs and photo ID work for providing both baseline data prior to disaster events and also reference data for comparison to affected areas. So we'll learn more about the overall impacts on marine mammals from the Deepwater Horizon oil spills from Ryan and Sylvain. But I'll start by basically sharing the role that we as a stranding network take when an oil spill incident occurs. So for those of you who are not familiar with us, I know we've probably worked hand in hand with many of you, but for those of you who are not familiar with us, I'll give just a brief introduction here of who we are and what we do. We were established in 1980, primarily to further the understanding and conservation of marine mammals. And this was really coming off the cusp of the Marine Mammal Protection Act of 1972, during a time when there was an increased interest in studying the animals that were stranding in the US along the coast. We're a nonprofit, 501c3, so fully independent nonprofit, which basically means we're supported by competitive grants and donations. And because marine mammals are federally protected under the MMPA, all of the work that we do is done under a formal stranding agreement with the National Marine Fisheries Service in order to respond to both live and dead stranded animals. So we work under that agreement to do the work that we do. The number of stranding networks in each state, of course, varies. But in Texas, the TMMSN, Texas Marine Mammal Stranding Network, is the sole stranding agreement holder for the entire state. And we work with other organizations, given such a large coastline. And several of those organizations are designees under our agreement. And that includes SeaWorld of Texas in San Antonio, and the Texas State Aquarium down in Corpus Christi. So We also collaborate, of course, with countless state, county, and city agencies and conservation organizations and other researchers. And we have a team of highly trained volunteers, some of which have been with us for decades. And they have a lot of specialized experience. So we wouldn't be able to do all of the work that we do without these people who make response, again, across such a large coastline possible. All right, so our primary headquarters is located in Galveston. We're set up to handle multiple animals for rehabilitation at a time if needed. And we also have a complete necropsy lab pictured here on the left, where we actually spend much of our time conducting a full suite of sampling on fresh dead animals. So some of the dead stranded animals with more advanced decomposition are typically recovered and sampled directly on the beach, where a lot of the state, county, and city agencies have become so important, particularly with carcass disposal. Once we've sampled an animal, that really helps with the human safety side, making sure that that's not compromised in any way once we've opened a carcass. So they're quickly buried and taken care of. Although SeaWorld of Texas is inland in San Antonio, they contribute their veterinary expertise and really experienced personnel during disentanglement and out of habitat response that we do. And that requires capture and a lot of experience from animal handlers. And they also provide that personnel and expertise during the critical phases of rehabilitations. We also work, like I said, closely with the Texas State Aquarium down here in Corpus Christi. So they have a wildlife rescue center with space for dolphin rehabilitation there as well. And that's been really helpful for live stranded animals in central and southern Texas, just so that animals can be stabilized sooner rather than having to make a trip all the way up to our main facility in Galveston to have them. We also work really closely with UTRGV, the Coastal Studies Lab in South Padre, near Brownsville, and they also have space where animals can be temporarily housed for stabilization prior to transfer for rehabilitations. Again, this collaborative effort is really useful, again, trying to cover such a large coastline. So one of our primary objectives on a continuous basis is responding to marine mammal strandings. So when animals strand alive, we aim to provide care for them, which may involve bringing animals in for rehabilitation, those that are good candidates for rehab. And at times it also means making the tough decision for humane euthanasia if needed. Response also includes rescue of entangled, out of habitat, or other animals in need of medical assistance. For dead animals that wash ashore, we aim to collect data and biological samples to learn more about life history, potential contributing factors of strandings, disease. There are a number of questions we try to answer when working up those carcasses. So on average, we respond to about 135 animals per year, and the majority of those are dead. Around 10% may be live stranded. So there's a lot of opportunity to learn from the carcasses that we receive. Another primary objective is to manage a long-term database for baseline values to compare across time and space. So baseline data serves as a starting point, really, that allows us to determine how much something has changed over time or after an event, such as an oil spill. So this established database has been really important for identifying unusual mortality events in Texas, whereby the data we collect can be quantitatively compared to these long-term data. We've been in operation, like I said, since 1980, so for over 40 years. So we have a large database, and our goal is really to continue building upon that work and manage that long-term information. So over the past 8 years or so, we've also been conducting bottlenose dolphin photo identification surveys. There are a number of stranding networks that get involved in this, but not every stranding network does. So for us as an organization that responds primarily to stranded bottlenose dolphins, having a better understanding of the free-ranging populations in Texas is really important to us. We collaborated with the National Marine Fisheries Southeast Fisheries Science Center, Pascagoula Lab, it's a mouthful, to conduct photo ID mark recapture abundance estimates for several of the Texas Bay, Sound, and estuary stocks of bottlenose dolphins in North Texas. And the TMMSN continues to conduct photo ID surveys to contribute to that established catalog, and to answer some more research questions that we're interested in that relate to occurrence, distribution, habitat use, site fidelity, the list goes on, but things that can really help inform some of our stranded animals. So we've also established a catalog for South Texas with work primarily in the lower Laguna Madre. So we're working to increase that catalog. These population studies and established catalogs are really useful for putting stranding data into context, basically for a better overall understanding of both living and dead dolphins that we respond to. So these catalogs also serve as an important baseline data, again, to compare potential changes to population dynamics, both during and after a disaster event. So our boat-based surveys also serve to document human interaction, such as harassment or entanglement, and to identify free swimming at-risk individuals that may be good candidates for early intervention that may otherwise go unreported. And finally, an overarching goal of ours is really to collaborate with other researchers with specialized expertise and to publish findings to advance science on all the data that we're collecting. This here is just a sample of recent collaborative papers we've published over the past 4 years, ranging from population abundance, behavior, and stranding trends, to novel medical procedures. So we really do work to get information out to the larger scientific community and also to engage with the public through talks and events where we set up booths. So we try to disseminate information as much as we can. We also, because we experience such a large number of strandings each year, we're also able to accommodate sample requests from various researchers. So we're currently collecting samples for quite a few researchers that have their own questions that they're trying to answer. Okay, so that's the stranding network here in a nutshell. In terms of responding to an oil spill, that will vary, of course, depending on the size and the scope of the incident and where it occurs. But essentially, responding to marine mammals is such a collaborative effort that can involve many institutions and organizations and different area researchers. So when we're first notified of a spill that has occurred in or around Texas waters, we quickly establish a plan with NOAA, and we're looped into the incident command system, essentially through a NOAA marine mammal representative for daily communication. There's a lot of communication that occurs during that time. And depending on the size of the incident, NOAA may also coordinate additional marine mammal responders to assist on the ground with response. We are a small nonprofit, and we have a lot of volunteers, but if the incident is very large, we may need support during those times. Okay, so one of our immediate goals, first off, in the event of a spill, an oil spill, is to identify and care for live stranded marine mammals that may be affected by the incident. So there are several pathways, of course, of oil exposure that can be toxic to marine mammals, and our goal is to locate and treat any living animals as quickly as possible. Annual HAZWOPER, or hazardous waste operations and emergency response. Certification is required for personnel handling potentially oiled animals, whether they are alive or dead. And during a marine spill response, we have staff that maintain certification year-round in the event of a spill. So we're ready to go. We're able to handle potentially oiled animals regardless of when that happens. So HAZWOPER certification can take quite a while for those of you who have done that sort of certification, you know, it can take many hours to get your initial certification and then to get your refresher training. So in the event of a spill, you want to move quickly. So we work really hard to stay up to date on our certifications throughout the year for that purpose. We also have staff that are trained in identifying oil exposure on stranded marine mammals. Searching for live marine mammals may involve a number of different survey types. So that may be aerial surveys conducted by TMMSN, NOAA, or other personnel. Aerial surveys are really time efficient, of course, and useful for looking for dolphins that are stranded or swimming in or near the oiled area, particularly in cases where the impacted area is either restricted or inaccessible, not easily accessible by foot or truck, or just a very large area. So where possible, land-based surveys are also conducted along the coast via ATV or truck. There may be several organizations on the ground searching for other wildlife too, so sea turtles, marine birds, that may also be affected and in need of attention. So at the TMMSN, we have marine mammal veterinarians that we work with to assess potential live stranded animals, and basically experienced handlers within our organization and designee institutions that can help collect animals. Our primary rehab center in Galveston is designed to handle multiple rehab animals at a time. And we also have the rescue center at Texas State Aquarium that can accommodate animals needed. Okay, so another primary goal in the event of a spill is to quickly recover dead marine mammals that may be affected by the incident. So it's important that we recover these carcasses as quickly as possible. Also, just so that the tissues are viable for testing, and also to avoid potential secondary oil exposure to other animals through things like predation, scavenging. We have coyotes and birds that often scavenge on marine mammal carcasses, and so we want to collect those carcasses and handle them as quickly as possible. And also, again, for human safety, just to get those potentially oiled animals off the beach. So the aerial surveys and land-based surveys used to search for live stranded animals, of course, can also serve to locate the dead animals in the vicinity. Given the potentially large and or remote area that may need coverage, local agencies such as city park boards and county road and bridge are really instrumental in either assisting with those land-based surveys or just notifying us of stranded animals in the event that they observe them during the course of their own work. So we've worked really hard to build those relationships over the years. So stranding personnel that maintain the current HAZWOPER certification can then, once we get those reports, we can then go out and respond to the animals that they've called in about. We follow protocol established by NOAA for identifying, documenting, and sampling marine mammals that have potentially been exposed to the oil, and all data records and biological samples that we collect are maintained under a strict chain of custody. And that just helps ensure the integrity of the samples and data, as well as their admissibility in any legal proceedings that may come from the event. So we use standardized forms, again, designed by NOAA, so that our data are consistent with other organizations and stranding networks within the northern Gulf of Mexico. And that standardization really helps when it comes to comparing across organizations. We also have veterinarians and staff that are trained in advanced sampling techniques. So when conducting necropsies, we have all of the parts, the biological samples, but also the carcasses are maintained. They're locked in freezers and they're kept in that chain of custody until the incident is formally closed. Okay, so after an incident, once waterways are unrestricted or as soon as we can gain access, boat-based photo ID surveys may be carried out, again, depending on each case. But this allows us to not only search for free swimming animals that may be affected, but we can collect data on again, occurrence, distribution, behavior, and individual animal sighting records that can then be compared to data collected prior to the event. Again, we're really working to grow our photo ID catalogs, both in the northern part of Texas and the southern regions, so that we have some baseline to compare to. So having these records is really useful to gauge if and how dolphins are using the affected habitat and if that has changed relative to the incident. Okay, so post-incident, once it's formally ended, we may move to a natural resource damage assessment step, what's called step-down sampling protocol. That step-down may still involve an elevated level of sampling beyond what we may normally collect, but we follow a protocol that will be updated over time on a case-by-case basis. Again, it's different for every event. So for example, we may take more samples from a carcass with moderate to advanced decomposition than we would typically only collect from fresh carcasses. So at this Texas Marine Mammal Stranding Network, we do work really hard year round to collect as much as possible. We do a full necropsy workup on all of our code 2 animals, and we try to maintain a really high level of collection as possible throughout the year, again, for those comparative data. We then work with NOAA to prioritize cases for analysis, and that may be based on different things. For example, stranding locations of particular animals, or animals that were visibly oiled when they came in, or based on some of the necropsy observations that may have been unusual, and so on. So there are different factors that go into prioritizing cases. We also utilize our long-term historical data, again, for those baseline comparisons to incident data. And our 40 years of collection over the years has really helped serve as a robust comparison. Okay, so the Texas Marine Mammal Stranding Network has contributed to several localized natural resource damage assessments over the years. And we do that through marine mammal stranding data collection and population surveys. So Texas was not actually included in the northern Gulf of Mexico bottlenose dolphin unusual mortality event that was associated with the Deepwater Horizon oil spill. You can see by this map here, the yellow shading footprint of the oil really terminates just east of the Texas-Louisiana border, and marine mammals in coastal Texas were not considered affected. Although we were not included in the UME, we were included in the NRDA process for all of the pelagic species associated with the Deepwater Horizon. There are no real borders in the ocean, and so there may have potentially been deep water animals that moved closer into Texas waters at that time. So we were included in that process for pelagic species. We were able to contribute to some of the research that came out during this time, though, and that is associated with the bottlenose dolphin UME, associated with the DWH. And we did that by supplying data collected simultaneously in Texas that were used as a reference group to compare marine mammals recovered within the impacted areas and the rest of the northern Gulf there. So papers from this contribution included a review of historical unusual mortality events. There's a demographic cluster analysis, and then a description of adrenal gland and lung disease observed in UME affected dolphins. So again, this reiterates the importance of a high standard of data collection and funding for analysis consistently year round, even outside of disaster incidents. So again, even though Texas wasn't included in the UME, we did collect data at a high level, and were able to use some of that data as reference for the impacted areas. Consistent data collection post-event is also really important. I should point out for identifying the potential long-term chronic impacts in addition to the acute immediate effects from the oil spill. So I've kind of talked a lot about our role during an incident and how we go out and collect live animals and carcasses. But we also, it's really important that we continue that collection throughout the year following an incident, again, because not every impact is observed right away. So in summary, stranding networks serve an important role in marine mammal conservation and restoration during oil spill incidents. We do that through rescue, recovery, and of course, providing comparative baseline or reference data from established databases. A lack of baseline or reference data can limit the full understanding of the extent that a disaster event has. And enhancing stranding network capacity and photo ID programs throughout the year will help in restoration efforts by increasing the survival of live stranded and entangled animals, by monitoring health trends over time, and by improving knowledge of wild populations and the threats they face locally. And that is my last slide. I will stop my screen share here and pass it back to Dani.
Wonderful. Thank you so much, Sarah. We appreciate it. That was great. We're getting a few questions in, so we're going to do all of our questions at the end with a question and answer panel. So keep putting your questions in our Q&A or our chat, and we'll make sure that we try to answer as many as we possibly can when we get there, get to that point in the presentation. So next, I would like to introduce Ryan Takeshita. He is the Deputy Director of Conservation Medicine and has been a research scientist with the National Marine Mammal Foundation since 2017. He served as the research coordinator for the Consortium for Advanced Research on Mammal Marine Health Assessments, also known as CARMA. So that was another group that was funded through this GOMRI program. He has helped lead the foundation's scientific contributions to a variety of natural resource damage assessments involving injuries to and/or restoration for marine mammals. So with that, I will hand it over to Ryan, and he'll talk about a little bit more of the oil toxicity, as well as some information on other vertebrates. So thank you.
Awesome. Thank you, Dani. So before I begin, I just want to mention that many of the publications and projects that I'll be talking about today can be found at our website at the National Marine Mammal Foundation, nmmf.org, or our consortium's website that Dani just mentioned, karma.org. And in addition to our research, you can also find a lot about our education and outreach efforts there as well. And during the Q&A session, we'll be joined by my colleagues, Lori Schwacky and Celeste Perry. And Lori led this consortium, and Celeste can tell you much more about all of those great education and outreach efforts. And just up front, even though I'm the one presenting to you right now, there were many, many people that contributed to this work. A lot of it was through Karma, as Dani mentioned, and that was funded by the Gulf of Mexico Research Initiative. Karma had researchers and professionals from over 20 different organizations that supported the fieldwork that we did, some of the expert elicitations, and some of the workshops that I'll tell you about. And I should say thank you to Sarah, because that was a perfect intro for this presentation. And so much of what we do, the research that we do, relies on all of the hard work done by the stranding networks. So thank you to everyone that helped make all of this research possible. So my talk today will be divided into 3 parts: a quick summary of what we've done before the Deepwater NRDA settlement. Then we'll focus on the work after that settlement and up to today. And then I'll give you some thoughts on where we can go from here. Now, most of the work that's been done has been with the dolphins in Barataria, in the Barataria Basin. But where possible, I'll try my best to mention work on other stocks of cetaceans that were affected by Deepwater. Okay, so let's start with a quick summary of the NRDA results. And Sarah just introduced it, so I don't think I have to redefine it for you. It went on from about 2010 to 2015. But I do want to mention that in parallel with that, there was a cetacean unusual mortality event. You might have heard of it as a UME, and that was determined to be caused by the Gulf oil spill. And that UME included over 1,000 stranded carcasses. And who knows how many more beyond that were just never found. Eventually, NOAA and the other trustees reached a final settlement, and that was accepted by the courts in April of 2016. So among the many, many things that we established during the NRDA was how dolphins are exposed during an oil spill. So first, by combing through survey data and photographs, it became clear really fast that despite previous assumptions, dolphins and whales did not avoid swimming in oil slicks, whether they be thick oil or thin oil. And since they didn't avoid the oil, cetaceans were likely exposed in a variety of different ways, right? So that could be inhalation of vapors and aerosols. It could be direct aspiration of liquid and droplets into the blowhole. It could be incidental ingestion as they're going through the water or eating prey. And then it could also just be uptake of chemicals through the skin. And because of these exposures, we saw a variety of health consequences. So that included lung disease, which was likely related to that inhalation and aspiration exposure pathway. We saw impaired stress response. In live dolphins, we saw low cortisol levels, their sort of fight or flight hormone. And in the dead stranded dolphins, there was a thinning of the adrenal cortex, the gland that's responsible for that activity. As we discussed with the UME, we saw increased mortality, and that was confirmed by many of the boat-based surveys that were done at the time. And we also saw decreased reproductive success. And that's again based on both those boat-based surveys, but also some of the ultrasound data that we collected during live health assessments. And these NRDA findings were summarized in a number of publications, and many of those are in this special issue of Endangered Species Research, which you can find. All right, so for the NRDA, we pulled together all of that information. We plugged it into a population model. And so we asked how the oil spill affected population numbers, this red line here with the dip in population size, compared to the theoretical population size if there hadn't been an oil spill. That's this black line up here that kind of holds steady over time. So this is how we can sort of quantify injury, right? The difference between these two trajectories. And we didn't just do this for the bottlenose dolphins in Barataria Bay, which this graph shows, but we also did it for the other marine mammal stocks that we thought were affected. And so that's off here to the right. Now, I won't go into the details here just yet. Don't worry, we'll talk about it more in detail later. But this was definitely one of the areas that CARMA was hoping to refine with our integrative modeling effort. Okay, so that's the super duper recap of what went into the NRDA. Now we'll transition to the research since then, including those Gomery and CARMA studies we've been talking about. And our goal for those is to follow up on whether these health and reproductive effects that we saw during the NRDA lingered beyond 2015. And we wanted to check whether some of the health effects that we saw in other species might also have affected marine mammals. And then we wanted to use all that new information to update those population trajectory models. And hopefully, apply these models to help inform restoration planning. Okay, but first we're gonna take a quick little detour to think about how we can use information about oil toxicity to other species to help us think about and understand oil toxicity in marine mammals. Because of course, toxicology research on vertebrate species especially protected species like marine mammals, it has a lot of reasonable hurdles, right? They can be ethical, they can be financial, they can be logistical. And so we have to find creative ways to figure out how oil affects vertebrate species and protected species. So as part of CARMA, and with additional funding from Gomri's Synthesis and Legacy program, we held a workshop to look at how oil affects any and all vertebrate species that were studied, with the hope that we could then apply those findings to species that weren't studied to the same extent or haven't been studied at all. So you can find a summary of our findings from the workshop in this paper in the Journal of Toxicology and Environmental Health, Part B. But our overall charge for the workshop was to take a multidisciplinary, collaborative approach to analyzing, interpreting, and synthesizing that wealth of data on the toxicological effects of oil, especially Deepwater Horizon oil, on vertebrate wildlife and humans. So in terms of that wealth of data, the biggest source was, of course, from the NRDA, which, in addition to everything I told you about with marine mammals, also, of course, included evaluations of oil's effects on other wildlife resources. That included birds, fish, sea turtles. And beyond wildlife, the NRDA also included a huge collection of controlled laboratory exposures. And what was especially important was that of those hundreds of laboratory tests that were run, they all use the same Deepwater Horizon oils. So we can directly compare the results within this testing program and then compare those results outside to what we know about exposures out in the Gulf during the spill. But we also knew that there was plenty of information beyond the NRDA. The spill resulted in a huge amount of laboratory and field studies, including different federal agencies, academics, industry, just so many different people following up their particular field of interest, right? And beyond the wildlife studies, we also had a bunch of folks looking at the effects of Deepwater Horizon oil on humans, whether it be the NIEHS, the Coast Guard, the Army Corps, the CDC, and then a variety of academic institutions. So we wanted to pull all of this together. So during the workshop, we brought together toxicologists, veterinarians, resource managers, and epidemiologists to comb through all of the literature and studies that we could get our hands on. And we wanted to find the consistent and inconsistent aspects of Deepwater Horizon oil toxicity across vertebrate taxa, right? So fish, birds, turtles, marine mammals, and humans. So one of our final products was a series of tables like this one, which you can find in that review paper. And so this is an example using the respiratory physiological system and the cardiovascular system. And these are two that were hit especially hard by Deepwater, we found out. So with the respiratory system, as in a lot of the situations, most of the cellular and molecular pathway effects were seen in fish, right? They're just easier to work with in the lab and do those sorts of tests. So we saw gill damage, we saw DNA damage, and we also saw effects on the fish's metabolic and aerobic capabilities. And then across some of the other species, we also saw evidence of asphyxiation, of inflammation, and in the dolphins, this sort of manifested as, lung disease via a variety of indicators. And then we also saw clinical indicators of respiratory disease in response workers and in coastal human residents. Similarly, there was a pretty impressive series of data that kind of put together a full pathway for oil's effects on the cardiovascular system. So starting with ion channel and cell disruption in heart tissue, that led to cell dysfunction, of course, and resulting cardiomyopathy. And then, we saw an increase in heart attack risk in humans. And even though I didn't update the table just yet, I'm about to show you some potential evidence in marine mammals as well. So in addition to those tables, we also tried to generate connected pathways, to show those connections between exposure and going up the biological levels of organization. So here's an example of the endocrine system moving from reasonable exposure pathways to molecular indicators like DNA damage and oxidative stress, to tissue-specific effects on the HPA axis, which result in clinical indicators like reduced hormone secretion, and then finally leading to whole organism and population level effects. And if you'd like to see other conceptual models for different physiological systems, you can go to that review paper's supplemental materials. So overall, as I hope you can tell, we saw some striking consistencies across taxa in terms of how Deepwater Horizon oil toxicity drove things like oxidative damage, cardiotoxicity, disruption of blood cells, immune system dysfunction, impairment of stress responses, and effects on locomotion. And so now we can jump back to thinking about, specifically how oil affects marine mammals, but now we can do so with the benefit from everything we've learned about how oil affects other vertebrate species, right? So in terms of the marine mammal studies that have been done since the NRDA settlement, as we have been saying, this is mostly done for our side of things in terms of how, in terms of this CARMA consortium. And so this is the framework that we took for our approach. And I'll step through each of these topics during the next few slides. So we'll start with the cardiac health assessment and the dietary analyses. I think Sylvain will likely touch on the immune aspects in the next talk. Then we'll talk about our evaluation of whether lung disease and reproductive effects continued to linger in the years since the settlement. And then finally, we'll review our integrative modeling approach. So part of CARMA wasn't just about doing the same things we've always done with health assessments, right? But we wanted to develop new veterinary and laboratory techniques so we can answer more questions about wild marine mammal health, especially for those adverse health outcomes that we saw in other vertebrate species, right? Like cardiac effects. So this is my colleague at the National Marine Mammal Foundation, Barb Linehan. She helped lead this cardiac part of the CARMA work. And so she helped our huge team develop techniques for auscultation. That's just listening to a heart with a stethoscope, with developing ECGs, echocardiography, and then helping to establish baselines for different histopathological lesion prevalences and also gross pathology effects. So we developed these methods and established these baselines using managed Navy dolphins. And then we applied what we learned to the wild dolphins. And so skipping over lots of fantastic work and lots of details, just to give you some of the most interesting findings here. Yes, dolphins living in Barataria Bay during the oil spill showed evidence of heart problems, just like a lot of the other species that we looked at with the TOV. So that included thinner heart walls, smaller heart chambers, and a higher prevalence of valve problems. Additionally, 2 Barataria dolphins were diagnosed with pulmonary hypertension. That's just an increase in blood pressure in the valve, in the areas connecting the heart and the lungs. So that could have been associated with chronic pulmonary disease. It could have been associated with oil cardiotoxicity. It could have been a combination of the two. But on that note, it's hard because these cardiac studies were done about 8 years after the spill. So it's hard to say for certain whether the observations are directly due to oil toxicity, whether it's indirectly through chronic issues caused by the spill with other diseases, or something else entirely. And most likely it's a combination of those things, right? All right, so now moving on to our assessment of whether Deepwater Horizon had an effect on dolphin prey, which in turn led to effects on dolphin health. So we examined stable isotopes and fatty acid patterns in dolphin tissues and prey species tissues from across the northern Gulf of Mexico. This work, I should mention, was led by Carl Cloyd and Ruth Carmichael at the Dolphin Island Sea Lab. And you can see 3 of the papers that resulted off here to the right. And while we didn't find direct evidence of prey shifts following the spill, the research was really useful in that we did show that stable isotope patterns, which can also tell us something about dolphin habitat use, can help to predict exposure to other threats like morbillivirus. That's a major disease for dolphins and whales. And we further found that isotopes and movement trends can help us predict a dolphin's relative resilience to environmental changes and to stressors like oil spills. All right, so now let's move on to some of the effects that we did know about during the NRDA. So including lung disease, right? And it turns out that based on our continued health assessments, yes, pulmonary disease did persist in Barataria dolphins at least 8 years after the spill. Barataria dolphins were more than 3 times likely to be diagnosed with moderate to severe lung disease compared to dolphins from Sarasota, Florida, which was an area that didn't get oiled from the Deepwater Horizon spill. And in fact, the highest prevalence of lung disease was in the final year of our study, 2018. And some conditions like alveolar interstitial syndrome, this is when liquid and cells fill the spaces that should be empty in your lungs. Some of these symptoms became more prevalent over time. And again, the highest prevalence was in our last year of study, 2018. So in general, the veterinarians established that pulmonary disease did contribute to poor overall health of the animals and negatively impacted both survival and reproduction as far out as a decade after the spill. Now, how about the decreased reproductive success? So to supplement those boat-based surveys, our team of vets, led by my colleague Forrest Gomez, developed advanced ultrasound techniques for looking at pregnant dolphins. And with these techniques, they're now able to detect fetal, placental, and maternal problems as early as the first trimester. And of course, that helps us determine the cause of reproductive failure, not just the fact that there was reproductive failure. And applying these techniques to the Barataria dolphins, the most common abnormality in failed pregnancies was an abnormal utero-placental unit. More than half of the failed pregnancies showed this abnormality, whereas only 1 of the cases in a successful pregnancy showed it. And then we detected a variety of other abnormalities that were only seen in failed pregnancies, never in successful pregnancies. And so overall, the vets are now focusing on these utero-placental changes for future health assessments. So in the context of the overall reproductive health, we saw that 96% of failed pregnancies in Barataria dolphins had evidence of maternal illness. That's compared to only 25% of successful outcomes. In terms of potential mechanisms, we saw that 65% of the negative outcomes in Barataria dolphins had that ultrasound evidence of placental dysfunction. And we could detect that as early as the first trimester. And then I didn't mention this in detail, but based on testing the carbon dioxide levels in dolphins' exhaled breath, we also think that pulmonary disease contributed to low-grade sustained fetal hypoxia. Okay, so to summarize the population health trends over time, the health of Barataria dolphins that were exposed to Deepwater Horizon oil hasn't improved, And in fact, it's likely gotten worse in the decade after the spill. This could be because dolphins are getting older. It could be because the chronic diseases that they have are getting worse. And it could be because these dolphins are now more susceptible to other stressors like diseases. We did have a workshop to look for evidence of new emerging stressors or an increase in other stressors in the area during that time. And we didn't find any evidence to strongly support that. So all of this is in contrast to dolphins in Barataria that were born after the spill. They seem to have health parameters that are in a normal range that you'd expect. The one exception were some of the effects on the immune system, and perhaps Sylvain is going to talk a little bit about that. All right, so as promised, how did we take all of that information and update these population models? So I won't go through all of the things that we did with the model. There were many, but one of the most important things is that because of all of these health data, we decided to add a cohort of animals that were exposed and never recovered. In the original model, we made the assumption that all of the animals would eventually recover. We also had an expert elicitation to determine what the proportion of those animals that never recovered versus that do recover was. And then we also updated some of the other underlying demographic and life history parameters. So letting the computer crank through the model, voila, here is the updated population trajectory specifically for dolphins in the Barataria Bay estuarine stock. So blue represents that theoretical, how many dolphins would there be if the spill had never happened? The x-axis is over time, years since the spill. And the model predicts that that number would have leveled off somewhere above 3,000 animals. And the width of this blue band represents the 95% confidence interval. The red represents the model predictions for how the oil spill affected the population size and will affect it out into the future. So you can see it sent it plummeting up until about right now, 2020, 2021, where we're below, the model predicts that the population is below about 2,000 animals, and then will eventually recover. Now, all of this is under the assumption that there is no restoration. And the hope is that the restoration activities will improve the right side of this figure so that the red trajectory comes up to the blue line sooner. So to put numbers to this, the model predicts that the oil spill caused a maximum decrease of 44% in the population size, that it will take 35 years for the population to get back to close to what it would have been without the spill, and that there were over 31,000 lost years of dolphin life in this stock. And this is just about to get published with all of the details of the model. So we are working on other species and other stocks, and those information are going to be coming out very soon. But here's sort of an example of for the years to recovery for a variety of species. And I won't belabor the conclusions, but suffice to say that our efforts for karma show that many of these oil-related disease conditions have persisted, and some have even gotten worse in the years since the settlement. So that brings us to the never-ending question, right? Where do we go from here? I know this presentation is specifically about oil toxicity and marine mammals, and so we'll talk about recommendations for things we should do before and during the next spill in the next few slides. But I just do want to take a moment to talk about how the Deepwater Horizon oil spill might continue to impact dolphins into the future beyond simply oil toxicity. Because although the trustees are working hard to design restoration projects that will hopefully benefit all of the affected resources, we just don't live in a perfect world where, we can benefit every single resource with everything we do. And sometimes that means the powers that be have to make some really tough decisions about, which resources are going to benefit from which projects. So in the case of Deepwater, one of the primary restoration projects that's been proposed is the Mid-Barataria sediment diversion. This is an attempt to combat coastal wetland erosion by diverting sediment from the Mississippi River into the Barataria Basin. But this will mean that high volumes of fresh water will also get flooded into the basin, and prolonged exposure to low salinity is not good for dolphins. And so based on our models, the models predict that the decreases in salinity driven by this potential project would lead to a drastic decrease, an 80 to 100% decline in dolphins in Barataria Bay. So basically the functional extinction of dolphins potentially everywhere but the barrier islands. So again, although direct oil toxicity has seriously impacted the dolphins, the overall fallout from the Deepwater Horizon oil spill, including this proposed restoration project, may have even more significant impacts on Barataria dolphins. Okay, so I'm running out of time. The work that I've shown you has had really important implications and established a lot of really great frameworks and techniques that we can use going forward for future spills. And, as Sarah mentioned, I think our work has demonstrated the importance of long-term, baseline monitoring of populations and health monitoring. But there's still a lot to be done, right? We still need to really keep thinking about these mechanisms. We need to think about how to refine our strategies for assessing smaller events and thinking about remote assessments. We need to think about how to apply what we've learned here to other species and to other areas, and how to integrate multiple stressors, how oil might impact important behavioral and social structures for marine mammals, and thinking about how to integrate this into response efforts like the one Sarah was talking about. And we know that other oil spills have already affected marine mammals since Deepwater Horizon, for example, in both Texas and California. So it's going to be really important that we continue to work on these with everybody's help. So thank you. That's all I have, and I can pass it back over to Dani and Sylvain.
Fantastic. Thank you so much, Ryan. We really appreciate you summarizing so much research that you and that whole team has really learned about. So next up, we are going to have Sylvain De Guise. He is a professor in the Department of Pathobiology and Veterinary Science at the University of Connecticut. His research interests focus on the influences of man-made and natural toxins on the health of aquatic organisms. These include marine mammals, fish, lobsters, and oysters, and a specific focus on the immune system. So with that, I will pass it over to Sylvain.
Thanks, Dani. And looks like I started with the end. Can you see, am I in presentation mode?
Yes, you are.
All right, thank you. It's always hard to follow Ryan, first disclosure. And Ryan presented a breadth of work that has been done in the last decade or so. And I'm going to focus on a little bit more depth and looking into the mechanisms and trying to convince you that what we found was real and really associated with oil. My lab focuses on the immune system. Although we work with a lot of very smart people, dedicated people from a lot of different institutions to be able to collect those samples and to integrate the findings with the other aspects of health that have been discussed. So an overview of my talk today, I'm going to start with a little bit of background. Ryan provided very extensive background, so I'm not going to go in any depth in there. We're going to look into what has been previously described as part of the immune changes in dolphins following Deepwater Horizon oil spill. We're going to look a little bit more depth in the mechanisms that are involved and what does that mean? What are the implications for that? So like Ryan explained, we spent much of our summers in the last 10 years with 80 or so of my best friends doing live captures in bottlenose dolphins to assess their overall health with a team of veterinarians, technicians, scientists, and we spent a lot of time not only generating results in our respective labs, but getting together and trying to put the pieces together and understand what that means. So a lot of field work, but also a lot of work in conference rooms or in front of our computers to try to integrate those findings across our different labs. We sampled dolphins as part of this study in Barataria Bay that was within the footprint of the Deepwater Horizon oil spill. And we collected samples in the same time frame from a reference population in Sarasota Bay that is probably the best studied population of wild dolphins in the world. And they were not in the footprint of the Deepwater Horizon oil spill. And our lab, like I said, is focused on the immune system. And one of the very important functions of the immune system is the ability of your adaptive immune system. So the immune system that is specific, recognizes antigens, and then responds to those antigens. The primary response of those cells is to get activated and proliferate so that you don't have one cell, but you have a lot more cells to respond to that specific insult. And one of the ways we mimic that in the lab is we take blood cells, we put them in a culture, and we stimulate them with mitogens, CONI and PHA in this case, to stimulate the proliferation of those cells compared to unstimulated controls on the left. And what I'm going to present today is a series of graphs that look at Sarasota dolphins, the reference population, the first column in blue, and Barataria Bay dolphins sampled over time. So in 2011, a year after the spill, is the first year we collected samples. And when we stimulate the cells of dolphins in Barataria Bay in 2011, they respond a whole lot more to the stimulus compared to dolphins in Sarasota Bay in blue. However, that increase in stimulation, in response to stimulation, appears to slowly fade off over time and return closer to normal in 2013 and 2014. So a change in the ability of cells to respond to a stimulus that appears to slowly return to normal over time. That was our first conclusion as part of that first paper. And when we compare it to the very few studies that look at experimental oil exposure, a study in mink found something that was very similar, that the response of T cells to a stimulation was increased over time after exposure to oil. So, and interestingly, about the same magnitude of increase in response. And a lot of people ask, is it bad if your immune system responds more? And as part of the National Toxicology Program, there's been a very extensive meta-analysis of tens of studies in lab animals. And the conclusion of that was that if you're responding more or less than anticipated, it's abnormal. And any abnormality in the response of the immune system can lead to changes that are not desirable. And a specific example of the ability of T cells of an increased response to T cells could have two consequences that could exhaust their response. So when they're stimulated again, they're less able to respond, or that could lead to in lab animals to other undesirable outcomes like autoimmunity. So when we continued to sample dolphins over time in 2016, 2017, and 2018, we find that we once again find an increase in the responsiveness of T cells to a mitogen, so that you stimulate them and they over-respond again. And that could mean one of two things. That could mean that dolphins got better and then got bad again, or it could mean that there was a steady increase in the responsiveness of T cells, but because we sample only a small portion of the population every time we do those live captures, that we might have missed the sustained increased responsiveness of T cells to a stimulation. But it's remarkable that it's so sustained in time and at about the same magnitude as we found in 2011. So we will at least call that a long-term disturbance of the immune system in those dolphins. And in order to verify that this was not associated with something else, we exposed mice to oil in the lab. And when we did that, obviously it's a much shorter time frame than we found in dolphins. But when we do reasonable exposure of mice in the lab, we found that there's not a real difference on the top between exposed mice in orange and control mice in blue. But interestingly, their offspring had a significantly increased response to stimulation of T cells in orange. So again, a mechanism of action that is consistent between dolphin sample in the field and very controlled experimental exposure in the lab, where there's nothing else that's different between exposed and control mice except exposure. So in trying to understand where does that come from, we looked at cytokine responses. Cytokines are small chemical messengers of the immune system. They're the means that cells communicate among each other, among different cell types, and sometimes stimulate themselves directly. And we find that there's some specificity to those cytokines. For example, on undifferentiated cells can either go on the left towards a Th1 response, and that is driven by a subset of cytokines. And that leads to a cytotoxic T cell response, so that the ability of T cells to destroy infected cells, for example, cells that are infected by intracellular pathogens like a virus. Or under different circumstances, different subsets of cytokines might lead on the right to a Th2 response. And that leads to the production of antibodies that will recognize and be effective at eliminating extracellular pathogens. So there's the other thing that's remarkable is that while they're complementary, they have a tendency to be a little bit selfish, and each response, in addition to enhancing itself, has the ability to inhibit the other response. For example, cytokines from a Th2 response directly inhibit the differentiated into a Th1 response, and as well, a Th1 response inhibits the further response of a Th2 response. And what we did is we measured those cytokines, those messengers that drive that differentiation in the blood of dolphins. And what we found is that the cytokines that drive a Th1 response in 2011 in Veterinary B were about half as low as in Sarasota Bay dolphins in, again, the control population. There were IL-12 was 20 times lower and interferon gamma was 4 times lower. So an overall inhibition in 2011 of the cytokines that drive a Th1 response, a cell-mediated response. On the other end, the cytokines that drive the differentiation towards the Th2 response were a little bit lower for the effector cytokines, but interestingly, IL-4, that is the cytokine that drives, the major driver of that differentiation of the direction of that differentiation was 3 times higher in Barataria Bay in 2011 compared to Sarasota Bay dolphins, and those appear to resolve over time. So the big picture is that there's a complete downregulation of the Th1 response that is responsible for cell-mediated immune response, and there's a mixed response, but mostly an increase in the Th2 response. And when we look in later years, we find that there's a continued suppression of the cytokines that are responsible for a Th1 response, and there's a continued increase in IL-4 that drives the differentiation towards a Th2 response. So an imbalance in those cytokines. And there's another cell type, they're called regulatory T cells. And those cells serve at dampening the immune response, inhibiting an over-responsiveness that can be damaging. And in 2017 and 2018, again, just like in 2011, those cytokines in the blood of Barataria Bay dolphins were much lower than in Sarasota. The other thing that we looked at, and Ryan hinted at that, is we looked at dolphins that were born after the spill. So they've never seen oil in their life, but their parents have seen oil. And we found that the dolphins that were born after the spill in Barataria Bay had Th1 cytokines that were much lower than the dolphins that were born before the spill, or who were alive at the time of the spill. And that is a little bit puzzling because, like I said, those dolphins that were born after the spill have never seen oil in their life. However, they might have been, their mothers might have been pregnant or might have been affected subclinically by the oil at the time of the spill. And we did not find that large increase in IL-4 in those dolphins that were born after the spill compared to those that were alive at the time of the spill. The Treg, the regulatory T cell cytokine IL-10, was again much lower in dolphins that were born after the spill. We did further studies to develop and validate lab techniques to identify regulatory T cells by the expression of FOXP3 that you see in purple here that you don't see when we use isotype control antibodies. So we can quantify those rare cells in the blood of those dolphins. And interestingly, we found about 3 times as many regulatory T cells in the blood of Barataria Bay dolphins than we found in Sarasota Bay dolphins. However, the cytokines that they produced were not 3 times higher. So that suggests that there's an overabundance of those cells, but they're not as functional. So the potential for a functional defect in those Treg cells. And again, very interestingly, when we expose mice to oil in the lab, and nothing is different, there's no other differences between the groups. The mice that were exposed to oil had over twice as many circulating Treg cells in their blood. However, there were no differences in the spleen. We went one step further, and when we looked at the not only what we find in the blood of those dolphins, but if we provide a physiological stimulus that promotes a differentiation of those naive T helper cells into either a Th1 response or Th2 response or a Treg response, do they have the same ability to respond if they're in dolphins from Barataria Bay compared to dolphins from Sarasota Bay? And what we found is when we stimulate the Th1 response, dolphins from Sarasota Bay, from Barataria, have about an equal response, maybe a little bit lower, but pretty much equal responsiveness to a stimulus to generate a Th1 response. However, when we stimulate a Th2 response, they're 2 to 3 times as responsive as Sarasota Bay dolphins. And that again fits the picture of a tilt towards a Th2 response in dolphins and vertebrate that would inhibit a Th1 response. And when we stimulate a Treg response, there's no difference between Barataria and Sarasota dolphins. And we push that one step further. And if we take Sarasota Bay dolphins T cells, we incubate them with physiological stimulus to generate a Th1 response. There's no statistically significant difference, maybe a very small increase with increasing concentrations of oil. But the Th2 response of Sarasota Bay dolphins that are exposed to oil in a culture medium demonstrates an increased responsiveness for a Th2 response, and there's no difference for Tregs. Again, if we put the pieces together, that suggests that oil, either in the lab or in real life in the Gulf of Mexico, oil exposure increases the responsiveness of T helper cells when there is a Th2 stimulation. So a pattern that is very consistent. So overall, what does that all mean? Barataria dolphins exposed to Deepwater horizon oil have modified T cell responsiveness, and the cytokines that direct the magnitude and the direction of those cells is also affected. It has a very consistent shift towards a Th2 response. And not only did we find that when we look at their concentrations in the blood of dolphins, but we also find that in lab experiments where we expose cells to oil. And those biases have been longstanding, almost a decade. The findings are also very consistent between dolphins that we sample in the wild and mice that are exposed in the lab. So that builds a weight of evidence that is very consistent and very supportive for the theory that oil is the responsible factor for those changes in the immune system. And what does that mean for those dolphins? Well, again, some meta-analyses performed by the National Toxicology Program in lab animals very clearly demonstrate that if your immune system is affected, you're going to be more susceptible to infectious diseases. So there's a very good chance that dolphins that swim in the Gulf of Mexico that encounter pathogens day in, day out, they're not always sick, but their likelihood of getting sick might be higher if their immune system is affected. And then the long-term aspect of those changes, including some aspects that appear to be more prominent in dolphins that were born after the spill, suggest that there might be continued exposure effects, continued exposure to oil in the Gulf of Mexico that drive those effects, or maybe that exposure stopped, but there's a potential for transgenerational effects, and that has been demonstrated with different contaminants in different species, including in our mouse study, changes in the proliferation of T cells that are found in the second generation that never saw oil. If that's the case, how long is that going to last? If there's effects that happen across generation or that are the result of continued exposure to oil, how long is it going to affect those? Is it going to affect those dolphins? And then what are the consequences for restoration strategies that might take into account the long-term recovery of dolphins? But if the second generation is affected, even if it's subtle, and those dolphins might still be young and might not have had the time to develop the long chronic effects that were found in their parents, how's that going to affect recovery over time? That's something that might be concerning. And I also would like to acknowledge the Gulf of Mexico Research Initiative that supported several of those studies, including the CARMA, which is probably the most stimulating consortium of scientists that I've worked with a very openness to collaborations and a diversity of results that Ryan presented earlier that are, I think, a hallmark of marine mammal studies when it comes to the effects of oil exposure. And with that, I will pause and take questions if there are any.
Fantastic. Thank you very much. We appreciate it. So we'll do a question and answer panel now. So if all of our panelists could turn on their cameras and their speakers. While we are getting set up, we've had a few questions coming in, and I just wanted to do a quick share of our publications. We have a number of different publications from our oil spill team here. One of the questions was about a number of ways to respond to dolphins and whale strandings. There's an app for that, and I would just like to highlight some of that information that we have for each of the Gulf states. So we've got that. We also have a marine mammal publication, but we have many other synthesis publications as well. I just wanted to briefly show you those, and then we'll get into that after our question and answer panel. So I think we've got everybody ready. So we have two other members, as Ryan mentioned, from the National Marine Mammal Foundation that are joining us. We have Lori Schwacke and Celeste Perry. So they will also be answering questions. They are very part of and very familiar with all of the research that has been discussed here. So I'll start off with, I guess, the question for, I think, anybody. Can oil exposure be detected non-grossly and later in time in any stranded individual, and how?
Yeah, that's a big one. I think we're still trying to work out, along with the broader toxicology community, what that sort of they call it a pharmacokinetic model of how the body takes in oil, and as it moves through different organs and into your blood and spreads out into tissues, how it changes over time. And I don't think anybody would say that we have a really great handle on it in vertebrates in general, nonetheless marine mammals. And so it's hard to directly detect PAHs or other oil chemicals in a body after time. So I think we're mostly dependent on trying to look for some of these oil-specific health effects that we were talking about. For example, the lung disease, or the adrenal cortex thinning. Some of the immune parameters that Sylvain mentioned are great because you can actually draw blood from a living animal instead of relying on a necropsy. So I think those are some of the things that we sort of rely on at this point to try to figure out exposure and effect.
Yeah, I would add that several of those chemicals that are in oil are broken down, so they're metabolized, so they don't stay forever. However, their effects may be long lasting.
That's a great point. Anyone else want to add to that? We've got plenty of questions. I'll keep them coming. Let's see, publications. Oh, so was satellite tagging acoustic telemetry data collected during the NRDA and Barataria Bay studies available to help interpret changes in the movement and habitat use elucidated by stable isotopes and some of the other data collected?
Yeah, you hit the nail on the head. That's exactly what we did. So you can look at those three publications where Carl was the lead author, and he worked with ourselves and Brian Balmer to do exactly what you're saying, to compare stable isotopes and look at dolphin movements to put all of those pieces together. And then in 2017, we also tagged dolphins further north in Barataria Bay to start asking how those dolphins might move in relation to low salinity effects, to start to answer some questions about that proposed diversion project. So that work is published in PLOS One this year, so you can find that.
Great, thank you. I have one for Sarah. What level of HAZWOPER training does your crew have? 24 hours, 48 hours? Could you tell us a little bit more about that?
Yeah, that's a good question. So we get the initial 24-hour HAZWOPER certification, which is primarily for individuals who will occasionally come into contact with hazardous materials, including oil, but we're not necessarily involved in cleanup efforts or consistently exposed. So it's just a little bit shorter than the 40-hour. And then each year we do an 8-hour refresher training. And it's really important for us to stay up to date and current on those for the work we do. And also 24-hour certification is really long and nobody wants to do that every year. So it's motivation to stay current. Yeah.
Absolutely. That's great. I'm going to kind of combine two questions here. And so how are groups like the Stranding Network, including Texas State Aquarium and SeaWorld Texas, paid for oil spill response work? And are there pre-established agreements or contracts?
So it's a very straightforward question with a very convoluted answer. So because disaster events are, require immediate response, there aren't always clear funds that are set up to be dispersed for that. But oftentimes, stranding networks or designee organizations may use their own resources initially, but then be reimbursed through contract or subcontract work through NOAA. I know NOAA does have emergency funds for some things like hurricane out of habitat response and some other things, but often it'll be the responsible party that reimburses that, again, through contract work that's later established. But initially from the moment it happens, sometimes that's just using the resources that you have in place already.
Thank you. Yes, never an easy question.
No.
We've got a few questions on little details, and I'd like to refer a lot of y'all to some of our publications about how you can contact Sarah and other stranding networks throughout the Gulf. It's very important, and we really appreciate everybody that falls into that. I have a question about restoration. So what restoration projects can increase population of dolphins so that they recover to the blue line? So they mean the blue line that Ryan was talking about. So how can we get those populations back up? And if you have any other restoration projects you'd like to talk about.
I guess I'll talk about this a little bit. Yeah, it's a really great question, right? And I think that everybody is working really hard in terms of not just Deepwater, but some of the other NRDA cases around the country to figure out what is the right scope for each of the events. So for Deepwater and for these other cases, one of the main things is helping people like Sarah do their thing in terms of enhancing stranding networks and trying to make those efforts more robust, more reliable, and get them what they need to and help us answer some of the questions we have for research, which in turn can be used to help those populations, right? But a lot of the efforts are also essentially trying to address other threats, right? So in areas with a lot of industry where oil spills are likely to happen, you also have other threats like fishing pressure. And so some of the restoration projects are aimed at, can we help disentangle animals more efficiently? Can we encourage programs with fishers to try to reduce the number of entanglements and that sort of thing? So those are just some of the ideas, but I would say that it's a hard question, and I think that a lot of the difficulty with marine mammals comes from the fact that restoration projects for NRDAs don't have a long history of including marine mammal restoration, and so we're sort of working in a framework of oiled marsh acres or fisheries impacts. And so now we're all trying to work together to figure out how to make the system work for marine mammals as well.
I would like to add to
Please,
that.
go ahead.
Reducing multiple stressors or additional stressors is certainly a way to avoid some delays in the recovery. And I'd like to put in a plug for Celeste and her group that have been a great job at educating the next generation, raising awareness, and I think raising awareness, reducing our reliance on fossil fuel will reduce the oil exploration and will reduce the number of spills in the future. I know it's not short term, but Celeste, maybe a word or two on the outreach efforts associated with the Karma efforts.
Yeah, absolutely. We worked hard to convey the information that was found through the Karma Consortium to a lot of different audiences. We tried to get it out to the scientific community, to the general public, doing webinars like this, but we really focused a lot of our youth outreach on working with the Girl Scouts and then other youth organizations. And we did some really fun workshops where we did a mock dolphin health assessment and walked the students through step by step all the things that happened during an assessment. And we shared some of the videos there that were part of the dispatches from the Gulf video series, which are still available online. You can actually see the process of going through one of these health assessments. And we also did a demonstration on how to care for oiled animals and how you would kind of clean up a spill. And the kids got to try cleaning up a spill and see how hard it really is. So yeah, hopefully we reached a lot of different audiences with that. And we know we got a lot of great feedback about our youth outreach stuff. And the great thing is that a lot of the information that we've developed working with Sea Grant and working with the GOMRI funding, we've directly translated to a program that we call Dolphin Doctor. It's basically all about marine mammal veterinary medicine and research. And so that's still available. If anybody on the line wants to schedule a virtual workshop with our group, then please let us know. You can reach us at outreach@nmmf.org.
Fantastic. Thank you, Celeste. Yes, we always think that outreach and education is incredibly important. And we love being able to work with all y'all. And we have many more resources online. I know we're sort of running a little bit over time. So if you can join us, we'll answer another question or two. If not, we please ask that you fill out the evaluation form. The link is in the chat, and that way we can get a little bit more feedback from y'all. We really appreciate that. I will also include, or I believe included in the chat is links to our oil spill team publications that we're able to share with everyone. Oh, there we go. The evaluation link is up in the chat box. So if you could do that. So let's do, I think we can do two more questions. And we have one for Sylvain. Is it possible that the immune response observed in the Barataria Basin has a genetic link?
It is indeed possible, but it's unlikely given that when we expose mice in the lab that all have the same genetics, we find very similar results. And when we take cells from Sarasota Bay and we expose them to oil in the lab in vitro in petri dishes, we find very similar results. So it is possible, but it's very unlikely, I think.
Good to know. And then double checking my questions. I think the last one is, how do you compare the degree and duration of exposure to the Barataria Basin dolphin population with wide-ranging oceanic whales? Is there a way to do that?
Sounds like a great one for you, Lori.
I was just waiting to see if you're going to fish it to me. Yeah, it's really tough, right? But what we did for the population models was basically look at surface oil maps, and we looked at the degree of oiling in Barataria Bay, and then also looked at oiling, the duration and the degree of surface oil in the offshore populations. And so we developed an index of as bad as Barataria, and that was how we tried to quantify the exposure for the offshore populations, assuming that given that exposure in Barataria Bay, that they would have the same effects. That is one thing, though, for future research. It's really tough to quantify exposure in these animals. We couldn't put little dosimeters on them to estimate what kind of exposure they're getting, but hopefully maybe in the future that is something we can do. We can get remote tags that can measure that sort of thing and really get a better understanding of what and how much animals are being exposed to. Just keep in mind, though, that these animals are swimming right at the surface where some of the most volatile chemicals from the oil are, and they're inhaling those chemicals. And so we think about the people's exposure that are out there doing cleanup. Well, the dolphins are there 24/7 every day and breathing right at the surface. So it is a fairly extreme exposure.
Great to know, and that's a great question. So that covers all of our questions. I would like to say a huge thank you to all of our speakers. I really appreciate your expertise and your time being here. I'd like to make a shout out to my comms communication team. They're doing all of the behind the scenes work, and I really appreciate that. If you could fill out the evaluation, we would very much appreciate your response and thoughts on that. And then finally, we will be posting, this is streamed to Facebook, so there'll be a copy on Facebook, and we will be posting this to YouTube as well if you ever would like to go back and reference it. We'll talk to the speakers, see if we can get some of their slides on our website as well. And if you have further questions or interest in information, please check out all these researchers' work as well as our oil spill publications. My last plug is we are sending out any of those publications for free. So if you are interested or could use those for a classroom or teaching, we would love to share them with you. So please, I'll put that link in the chat as well. But we're trying to share as much of this wonderful information that everyone has done over the last 11 years. So with that, I will say thank you, and thank you all for joining, and I appreciate your time.
You.
Thanks, everyone.
Dani.
Thanks,
Thanks
Dani.
for hosting.
Absolutely. Bye, y'all. Have a great day.